what makes the Telaprevir, am I saying that correctly?
Yeah.
Telaprevir. What makes that different from other hepatitis C drugs on the market?
There's 2 hepatitis C drugs on the market right now.
Okay.
Both of them are not, well, one of them is pegylated interferon, and that's a biological molecule. It's not a small chemical, but it's a protein drug. And the second drug is called ribavirin, which is also a small molecule. So the protein drug you have to inject once a week, and the ribavirin drug you have to take by mouth. And you take them 2 together. And for most people in the United States who are genotype 1, there's many types of HCV virus. The typical treatment is 48 weeks of taking this combination of drugs, the ribavirin every day and the pegylated interferon every week. 48 weeks of this treatment. And then after 48 weeks, if you're undetectable, you've driven the virus, cleared the virus from your bloodstream, you wait another 6 months. 24 weeks to see if you're still negative, and then you're considered more or less clear. That's the standard of care, and on average, people, about 60% of the people who take it fail the treatment. And it also makes you feel terrible because interferon is what your body naturally makes when you get the flu or any kind of virus. So these people feel like they have the flu for an entire year, and many of them can't hold on to drugs. The ribavirin gives them anemia. It's just a really, really difficult treatment. So the way Telaprevir differs from those is the treatment that we have gone into and just completed our phase 3 studies, in the sense that we completed dosing for phase 3, is a combination treatment with another small molecule called Telaprevir. And that molecule inhibits the virus directly. It inhibits an enzyme called the HCV protease. What happens is the HCV, when it goes into your body and infects you, it creates this very long string of protein. It's a very, very long polyprotein. And in order for the enzyme for the virus to make more virus, the protease has to clip between different portions of the polyprotein. If you knock that out, if you inhibit that, the virus can't replicate. So when we combine that drug with the standard care pegylated interferon, we can treat patients for half the amount of time. Instead of 48 weeks, it's 24 weeks. It's 12 of the triple plus 12 of pegrivid. And we have greater than 20% improvement in the SVR rate, so a higher cure rate. So that's both in the length of treatment. It's a new drug. It has a different mechanism of action, and it has a higher SVR rate. Those are all new things. And right now, where we are is we've just finished phase 3, the dosing. The last of the 3 major steps that you have to go through to get your drugs approved in the United States and Europe. So there's phase 1, 2, and 3. At this site, with the IRA group, so my group, I'm in charge of infectious diseases, we do, as Uta told you, both antimicrobial and antiviral. For the clinical trials, we have to monitor whether the patients who don't get an SVR, if they become resistant to the drug. And so there's people here who are actually really the people who are doing the sequence analysis. And they work very closely with us in Cambridge, Massachusetts, and we go back and forth. And so they're involved in putting together data and analyzing that actually goes to the FDA. It's part of what's called an NDA, a new drug condition, an application for the new drug. So we're very, very closely integrated.
All right, and so this new drug, this is something to take along with the current treatments.
Right now, that's what it is. Yes.
Okay, and again, could you just sort of explain? You said it prevents the virus from replicating.
Yes.
So
do you want to understand kind of how it works?
Yeah, how that works.
HCV virus has genetic information. It's actually RNA. Some viruses have DNA, some viruses have RNA. This one happens to be RNA. That RNA is a code that can be translated to make a long protein, which by itself is not very functional. Part of that protein is cleaved. It can make more virus. It can make the coats that basically is like the car that surrounds the driver, which might be the RNA. The rest of it makes the enzymes that helps to make more copies of the RNA. And HCV protease is an enzyme that cleaves between adjacent amino acids, releasing separate proteins that are part of this long chain originally, the long polyprotein. And in order for those proteins to really work, they have to be cleaved and they have to come together. So what it does is actually stops the replication cycle right there. And you can't get any more little HCDs. That's how it works.
All right, and how does this how does moving to the BioVenture Center help with this whole process?
Well, I mean, just very simply, it's a lot more space.
Okay.
We were really, really crowded in the other site. I think what it mostly does is it gives us a little bit more room and opportunity to expand what we're doing here in Iowa, which I hope we'll be able to do when Vertex becomes profitable. Right now, we're not profitable, and telaprevir will be the treatment of telaprevir plus pegrivi will be our first major drug. So it really gives, in my view, I couldn't imagine expanding the group in the cramped space that we had before, which was an incubator space, actually, to begin with. So this, to me, is a commitment from Vertex to the community here and to the group here that, indeed, this is a really good group, this is a great space to be in, and this kind of atmosphere is the kind of place where we want to do more. So that's what it says to me.
Okay.